Thank you very much for inviting me here. It is a challenge to talk about systemic therapy in mesothelioma. We don't have that many options, but we will try to go through the latest things that are available, and specifically we will go through the immune therapy. I also have no disclosures. So the only therapy that is currently available to us is chemotherapy, and the only FDA-approved therapy for mesothelioma is pemetrexed and cisplatin. This was a kind of a landmark study that put this chemotherapy on the map. Pemetrexed and cisplatin was studied versus cisplatin alone in a randomized phase 3 clinical trial. 456 patients were looked at. They were randomized to two groups and randomized to cisplatin pemetrexed versus cisplatin alone. Oops. And this is what the study showed. The response rate was dramatically higher in the combination group. It was 41% versus 16% in cisplatin alone. And the study established the combination of cisplatin pemetrexed as the standard of care. The current recommendation is to administer at least 4 courses, and if the delayed response is seen and therapy is tolerated, 6 courses can be administered. Can carboplatin be substituted for cisplatin? And basically the answer is yes. And this was shown in a multicenter, non-randomized, open-label trial in chemotherapy-naive patient, but showed a pretty similar response rate in patients treated with carboplatin versus cisplatin. So cisplatin and pemetrexed is still the regimen of choice. So given that, we're looking for other options, and of course, we're now turning to the immune therapy. There has been a long-standing interest in turning on the immune system to treat cancer. The first real data was shown in 2007 by Coble that demonstrated that tumors that were induced in mice by a carcinogen could be controlled by the immune system. And what we saw is that suppressing the activity of the immune system allowed dormant tumors to escape from the control of the immune system and start dividing, disseminating, and eventually cause death of the host. As it turns out, this was looked at much earlier. And in 1909, Ehrlich introduced the concept of tumor dormancy, and for which he went on later to win on the Nobel Prize. So now immune surveillance concept is accepted by the scientific community, and avoiding immune destruction is actually included in the hallmarks of cancer. These are the hallmarks of cancer, and this, of course, was introduced by Weinberg in the 1990s, and those are the concepts and the organizing principles for rationalizing complexities in the neoplastic disease. So the original hallmarks included evading growth of cancer, activating invasion and metastases, enabling the tumor cells to replicate and be immortal, induction of angiogenesis, and resisting of cell death. So the two new emerging hallmarks that were added was reprogramming of energy metabolism and evading the immune destruction. And you can see that. You can see these new concepts that were added over here. So immunotherapy is now considered to be a third wave in cancer treatment after conventional therapy and targeted agents. And unfortunately, targeted agents are in development, but we don't have any specific targeted agents in mesothelioma. Immune surveillance is an essential mechanism in the prevention of cancer developing and is dependent on the immune system recognizing tumor antigens as foreign and being able to eliminate them. So this is the immunoediting hypothesis. Immunoediting is thought to occur after intrinsic tumor suppressor mechanisms have failed and neoplastic transformation has begun. So this is the normal tissue, and this is where the immune cells are introduced, and these cells become transformed. So the first phase is called elimination phase. It consists of host innate and adaptive immunity working to destroy the subclinical tumor. You have seen this in the earlier presentation where multiple cells of the immune system, the CD-positive cells, the tumor-infiltrating lymphocytes, all play a role, and they try to eliminate the tumor. Cancer cells that survive this phase subsequently enter equilibrium, where growth and metastases are constrained by the competent immune system. So, and this is again always taking place in normal hosts. However, when a resistant tumor variants emerge, they can evade immunity through resistance to immune cytotoxicity or induction of immune parasites in the host, and we will review all these agents as we go on in our talk. So what are the strategies that we have that can activate the immune system? There are several of them. First of all, we have monoclonal antibodies to immune checkpoint inhibitors. So antibodies that prevent the deactivation of the immune system. Immune conjugates is also considered in the category of the monoclonal antibodies, and it's Pseudomonas Immunotoxin. We have biological response modifiers that again either turn on the immune system or slow down the brakes of the immune system. Cellular immunotherapy, and we will talk about the chimeric antigen receptors, and we will also talk about vaccines, and the one vaccine that is currently in developing is the live attenuated Listeria monocytogenes vaccine. So how does the immune system work? It starts out with antigen presentation. So if this is the tumor cell, these are the tumor antigens. They're presented to the antigen, to the dendritic cell or to the antigen presenting cell, and they're displayed on the surface of the antigen presenting cell together with a major histocompatibility complex. So the immune system knows to recognize these antigens in the concept of self. T cells generate an immune response by binding to a stimulatory receptor, which is called B7, which is on the antigen presenting cell, via the CD28 receptor, which is on the T cell. And this step, as you can see right here, augments the excitatory antigenic stimulation that leads to T cell stimulation, activation, and potentiation of the immune response. There is, to avoid the overactivation of the immune system, there is an increased expression of the cytotoxic T lymphocyte-associated antigen, or CTLA-4, which is a transmembrane protein. So what CTLA is expressed about 48 hours after the T cell activation, and it competitively binds to the B7, which is located on the antigen presenting cell, outcompeting stimulatory CD28 receptors. So CTLA in normal tissues prevents autoimmunity and prevents the overstimulation of the immune system. And CTLA-4 binding provides dominant negative signaling and inhibits the immune response. So there is a rationale for CTLA blockade, which is called releasing a brake for CTL activation. So antibody to CTLA-4 prevents interaction with the B7 and removes the downregulation of the immune system. And the blockade of this checkpoint provided the first evidence of improvement in overall survival in patients with metastatic melanoma, which originally was brought into clinical practice in 2008. CTLA-4 inhibition is the furthest agent that has gone on in clinical trials in mesothelioma. There are two antibodies that are available. Both are fully humanized monoclonal antibodies. One of them is ipilimumab, and it was approved by the FDA for the treatment of metastatic melanoma. And the one that is currently studied in mesothelioma is tremelimumab. Tremelimumab is a fully humanized monoclonal antibody. The half-life is about 20 days. And again, it blocks the inhibitory signals that are provided from the T cell. It enhances the T cell activation and proliferation, and it enhances the anti-tumor activity. It has been used as a single agent in 9 clinical trials. And the first study that sort of put the tremelimumab on the map was a study from Italy, which just recently was updated at the IMIG meeting in South Africa in October of 2014. And this is a phase 2 study of the anti-CTLA monoclonal antibody tremelimumab in chemotherapy-resistant advanced malignant melanoma patients. So these are the patients that have already been treated with chemotherapy and that have progressed. 29 treated patients were involved in the study, 21 epithelioid, 6 biphasic, 1 sarcomatoid, and 1 patient's histology was undefined. The study met its primary endpoint at 16.5 months. 4 of the patients achieved a partial response, and 4 of them achieved stable disease. The disease control rate was 51%, and overall survival was 11.5 months. The most common adverse effects that we've seen are the effects that we see now with immune therapy agents. They're different from chemotherapy, and these are mostly the skin, gastrointestinal effects, and fever. The study was expanded, and we were a site. We participated in this study. This is a study that called Determined. It was a phase 2 randomized double-blind study comparing tremelimumab to placebo in second or third line treatment of subjects with metastatic mesothelioma. The study was very large and enrolled very quickly. It enrolled 564 subjects who were randomized 2-to-1 to get tremelimumab or placebo. The primary objective was overall survival. Secondary objective was progression-free survival, durable complete response, biomarker analysis, and validate quality of life questionnaires. So what we have from our experience at UCLA, what we have seen is that the patients that have been enrolled in the study, the ones that responded, stayed on the study. And in fact, the study was started about 2 years ago. Several of the patients are still on the study and receiving therapy. So responses have definitely been durable responses. So as I showed you, this is the CTLA pathway where the antigen-presenting cell presents the antigen together with the MHC complex, and the CTLA pathway is activated. Another pathway that is currently being investigated is a very active pathway called PD-1 complex, PD-1 pathway, and PD-1 is another protein in regulating the immune response. CTLA-4 and PD-1 are examples of what we call immune checkpoint proteins. These are co-inhibitory proteins which specifically diminish antigen-specific immune response. And PD-1 is named for its involvement in programmed cell death. And like CTLA, it's expressed on activated T cell and upregulated upon stimulation. And again, while it's undetectable on the surface of the resting T cell, it is found on the surface of the activated T cells within 24 hours. PD-1, what is interesting about PD-1 is that the ligands for PD-1, PD-L1, and PD-L2 are expressed on cancer cells. So in this way, this pathway is co-opted by tumors through the expression of these ligands. So this allows the tumors to specifically turn off the immune system and to turn off the anti-tumor cytotoxic activity of the immune system. So the first anti-PD-1 antibody originally was approved in melanoma, but also the agent nivolumab was approved for patients with advanced squamous cell lung cancer that have progressed on platinum-based therapy. This approval took place in March of 2015, and this was based on a trial which was called Checkmate trial, in which we actively have participated at UCLA. And it's a phase 3 open-label randomized multinational multicenter trial in which nivolumab was compared to docetaxel. 277 patients that have been previously treated with non-small cell lung cancer. Patients were randomized to nivolumab alone versus docetaxel. The median overall survival was improved with nivolumab. It was 9.2 months versus 6 months in the chemotherapy arm. This was considered to be improved overall survival by 41% with hazard ratio of 0.59 and a p-value of 0.0025. Again, at UCLA, we have studied another antibody, and the study was just published in New England Journal of Medicine by Dr. Geffen from the Oncology department at UCLA. And they have demonstrated safety of anti-PD-1 antibody pembrolizumab, which is in the same class of antibodies. And what was distinct about this study is that they have done a biomarker analysis where they clearly demonstrated that the expression of PD-L1 at 50% was correlating with the clinical efficacy. And the patients that had the higher PD-L1 expression had a much better survival. So pembrolizumab is of interest to us because this is the trial that is currently in development in patients with malignant pleural mesothelioma. The trial has not been open yet, but is supposed to be opening soon. So these two antibodies have different pathways. So as in other solid tumors, there is data that combination of these two antibodies can be useful. And one such abstract was presented in the IMIC meeting in South Africa. This was only a preclinical model, but nevertheless, the combination of the anti-PD-1 and anti-CLA4 together with cisplatin was found to be effective in delaying tumor growth and improving survival. The next drug that we have available to us is Pseudomonas Immunotoxin. Mesothelin is a tumor-differentiated antigen that is preferentially expressed in epithelial malignancies, including epithelial and biphasic mesothelioma and ovarian and pancreas carcinoma. So this is the immunohistochemistry showing the staining for mesothelin. The immunotoxin is a recombinant protein that links the variable fragment of the antibody with a portion of the Pseudomonas exotoxin. And in early phase 1 trials, the toxin showed some anti-tumor activity, as you can see demonstrated by the clinical picture and by the scans. So despite the initial promising results, the use of immunotoxin was limited by the development of neutralizing antibodies. And Dr. Hassan, who actually presented his data for the first time at this symposium, showed that a strategy of using two chemotherapeutic agents, pentostatin and cyclophosphamide, was effective in eliminating the antibodies. So what was more unexpected is that there was also significant anti-tumor activity in heavily pretreated patients that had poor prognosis. So what felt to be the mechanism of this, it was not only the antibody elimination, but it was felt that the cells of the immune system that were downregulating the immune system were eliminated as well, and especially the cells that were implicated were the T regulatory cells, which were eliminated by the addition of the pentostatin and cyclophosphamide. These are the results of the study. This is the waterfall plot and the scans. 3 out of 10 patients with extensive tumor burden had a durable partial response. Again, 2 patients who did not initially respond to the immunotoxin had a dramatic anti-tumor response several months later, suggesting that pentostatin and cyclophosphamide could have had an effect on upregulating the immune system. And antibody formation was markedly delayed. They went on to study the immunotoxin together with pemetrexed and cisplatin in the first-line setting, and they had a 60% partial response in 3 patients that had stable disease. So this study is currently ongoing for the patients with mesothelioma in the first-line setting where immunotoxin is combined with chemotherapy. The chimeric antigen receptors is where the 21st century medicine and gene transfers come in. This is very exciting and is probably going to be the hottest area in medicine. This is another approach of targeting tumor-associated antigen. So chimeric antigen receptors, or CARs, are comprised of 2 parts. They're comprised of the tumor antigen binding domain, which is right here, that is linked to an intracellular portion of the T cell receptor. So they unite the high-ability antigen recognition with T cell signal transduction. And the antigen binding, so the antibody is finding the antigen, and then it activates the signaling cascade that induces antigen-specific cytolysis, effector cytokine and cytokine secretion, and T cell proliferation. This is a complicated way of doing this, similar to pheresis and procedures that are done in stem cell transplants. Cells are collected from the patient by apheresis. The T cells are subsequently expanded and grown in large numbers, and then the chimeric antigen receptors are transduced into T cells via lentiviruses. And this is where the actual gene transfer takes place, and then the T cells are controlled and subsequently returned to the patient. So actually there was a phase 1 clinical trial, and also was presented at the IMIG meeting in 2014 in South Africa. This was the first phase 1 trials of anti-mesothelin mRNA chimeric antigen receptors in patients with mesothelioma. Patients, again, had progressive disease after cisplatin pemetrexed therapy. They all had apheresis, ex vivo T cell expansion, and electroporation of the cells with the CARs. Their primary outcome was safety, and secondary outcomes measured radiographic response, T cell trafficking, and generation of the secondary immune response. These are just some very preliminary results. They have studied only 7 patients. 1 patient had a sustained response at 2 and 3 months. Another patient had a mixed response at 2 and 3 months. All other patients had stable disease. Unfortunately, at 6 months, all patients had progressive disease, and 1 of the patients has developed antibodies. So a new study is currently undergoing, where they're also using immune conditioning, similar to what I have showed you with immunotoxin, where they're using lymphodepletion regimens to get rid of the antibodies, and the study is currently open through the NCI. This is the data showing the vaccine. CRS-207 is a live attenuated Listeria monocytogenes vaccine. This vaccine is engineered to express tumor-associated antigen mesothelin, which, of course, as I showed you before, is highly expressed in malignant pleural mesothelioma. The CRS-207 stimulates innate and cellular immunity. And again, this is coming from the NIH. Hassan and colleagues have a trial of the CRC-207 vaccine together with chemotherapy in the frontline treatment of patients. And the way it's done is that they receive 2 vaccinations with CRS-207 2 weeks apart, followed by 6 cycles of chemotherapy, 3 weeks apart of standard chemotherapy. And then this is followed by boost vaccinations 3 weeks apart. If the patients are clinically stable, they could continue on maintenance CRS-207 vaccinations every 8 weeks. They have not observed any adverse events. And out of the 16 patients, they were able to show a 60% response, and 27% of the patients that had stable disease. And this is better than what we see with chemotherapy alone. So what I try to show you is how the immune system can be targeted. It can be targeted through antibodies. It could be targeted through CARs, through vaccination, and through cellular immunotherapy. So hopefully this is going to be the 21st century in immune therapy and mesothelioma. Thank you.